In this video, we are going to continue our discussion on protein synthesis. We have now created the messenger RNA, and it has exited into the cytoplasm. So what happens next is something called translation. Just a quick review on some of the things that we've learned. We know that nucleotides are the monomers of DNA and RNA, and the nucleotide is either A, C, T, or G in DNA, and for RNA it is A, C, U, or G. We know that amino acids are monomers of proteins, and there are 20 different amino acids that makes up all the proteins we know in the body. So how do we decode the mRNA? You should know by now that the mRNA is actually a copy of DNA, and that segment of the DNA is a gene that contains instruction for making a particular protein. So it turns out that in the DNA code, three consecutive nucleotides makes up what we call a codon. And so here you can see ACC is a codon, and when you copy it in the form of mRNA, it's going to become a UGG. Because if this is the template DNA strand, then the mRNA is going to be complementary to it. So UGG is a codon on the mRNA, and each codon is going to encode for one of the 20 amino acids. So in here you can see UGG encodes for TRP, which is a tryptophan, an amino acid. So there are going to be a total of 64 codons. Okay, so a codon is 3 nucleotides, right? So the first nucleotide, you have 4 options. It can either be A, C, G, or U, and the second one also 4 options, and the last one also the same 4 options. So if you do 4 times 4 times 4, that's going to be 64. So a total of 64 different codons are possible with 4 nucleotides. And these 64 codons are going to be coding for 20 amino acids, which means you have more codons than there are amino acids. So some codes are going to be redundant. Right? You're going to have multiple codes coding for the same amino acid. And that's actually a good thing because it offers some wiggle room in terms of mutations. Later on in the lecture, we're going to look at what happens when the DNA changes as a result of mutation. Because you have redundancy in the codons, sometimes changing the DNA might not necessarily going to be resulting in a different amino acid, and that's kind of a good thing. It's a protective mechanism. So of the 64 codons, there is one special codon called the start codon, okay, which is AUG. Right? All the proteins are going to start with this start codon. That's how the ribosome knows where to go to and begin the process of protein synthesis. So AUG is the start codon, and then there are going to be 3 stop codons. Okay, UGA, UAA, UAG. So this tells the ribosomes to stop the process of protein synthesis. So to remember these, you can think of this memory aid. Right? So you got A in biology. Right? So you are awesome, right? And you are good. Right? So that's how you can remember the stop codons: UGA, UAA, and UAA. UAG here for the last one. So here is a codon chart. The codon chart will help you decode the codons without memorizing all the codes. And you want to have a codon chart next to you anytime you do a quiz or a test. So you will be able to do questions related to translation. Here is an mRNA sequence, right? AUG, blah, 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 blah. So we're going to use the codon chart to decode this. So this column represents the first nucleotide, and then this will be the second nucleotide at the top, and then over here that will be the third nucleotide. So if I'm looking for the amino acid that is encoded by AUG, right, which is the start codon, by the way, we would look for A here and then U. So A and U means we're going to be looking at one of these four things. And the last nucleotide is G. So G is the last one in this box. So that codes for MET. Okay, so these are short forms for the amino acids, right? MET is methionine. You don't have to know the full names, but just know that they are actually short forms. Okay, so the first amino acid is going to be MET, right? And the ribosome will connect the MET to the next one. So it will read the codons AUU, right? And A is here, U is here, and U for the third one means it's going to be ILE. Okay, so isoleucine, that's the next one. And then the next one is CCU. Right, so CC and then U, so it's going to be proline, PRO. Okay, so try to do the rest before you watch the video. CAU, CAU, it's going to be histidine, HIS. Next one is CGC, CGC is going to be arginine, ARG. And then UAU, UAU, it's going to be tyrosine. UUC is going to be phenylalanine. UUU is going to be the same thing. And then we're going to have UAA, and you might remember from the previous slide, UAA is going to be a stop codon. So then the protein synthesis process is going to be completed, and you will have a protein that has 1, 2, 3, 4, 5, 6, 7, 8 amino acids in it. So that's how you decode the mRNA and figure out what amino acids are needed to be put together to form the protein. So here is another example. Right? Decode the following mRNA sequence into amino acid sequence. So you can go ahead and try that on your own, and then you can check the answer afterwards. So AUG, right? AUG is going to be MET. GGA, GGA is going to be glycine, GLY. And then the next one is CAU, so CAU, it's going to be histidine. UAU, UAU, it's going to be tyrosine. UUU, UUU, it's going to be PHE. GGG, GGG, it's going to be glycine. Next one is GCG, which will be alanine right here. And then UGA is a stop codon. Should be pretty straightforward, I hope. As long as you have the codon table, this would be quite easy. Example 2. Okay. Now we are given a template strand, and we are going to have to make the protein. All right, so that means we actually have to do the transcription step ourselves, right? So we have to make the mRNA first. So from the template strand, we can create an mRNA by using DNA RNA base pairing rule. So T is going to pair with A is going to pair with U, G, C, A, U. This one is going to be GGA. Next one is UUA. This one will be UGU. And this is CCG. And finally, ACT will become UGA. So assuming this one is all exons and we're not removing anything, then we can produce our protein. Okay, and you can always make that assumption. Okay, assume that this is the entire coding strand. So the proteins now, AUG is a start codon, and it's always MET, right? CAU is going to be histidine. Hopefully you're more familiar with the chart now. GGA is going to be glycine. UUA is going to be LUU. UGU is going to be cysteine. It's kind of running out of space here. So CCG is going to be proline, and then UGA is going to be stop. Okay, so that's how you make the protein. Here is another example. This time we are giving you the coding strand as opposed to the template strand. So the quickest way to get the mRNA from the coding strand is just to copy everything, but you replace the T with U. So this is AUG, ACA, UUA, CUA, GCA, GGC, ACU. Okay, so then you follow the same thing. Okay, so AUG is going to give you the MET. ACA is going to be the THR. UUA is going to give you the LEU. CUA it's going to give you LEU as well. GCA is going to be ALA. GGC is going to give you GLY, and then ACU is going to give you THR. And you notice there's no stop codon at the end. Remember, in reality, the proteins are actually sometimes made up of hundreds and hundreds of amino acids. So this could be just part of a gene, right? This could go on for some time, right? So don't worry too much if it doesn't end with a stop codon. Okay, so go through these 3 examples again if you need to, and make sure you understand them. So to the actual process of translation, in terms of how the ribosomes are gonna be able to decode the RNA, well, there are 3 types of RNA that are involved in the process of translation. We already know the messenger RNA is going to be the one that's going to be containing the actual instruction, the codon, that tells you what amino acid to put together. The ribosomes is actually made up of something called the ribosomal RNA, or rRNA, and the ribosome is important for creating the link to connect the amino acids together. That's the one that's going to be creating the peptide bond to link the amino acids together. Whereas the transfer RNA, or the tRNA, is the one that will be bringing the correct amino acid to the ribosome so that it can link it together. So you can think of this process as like, cooking with your kids or something. There is going to be a cookbook, the mRNA, that tells you what ingredients to put together to make a cake or something. You are going to be the rRNA, the one that's actually going to be making the cake. And then your kid is going to be the tRNA, who will be helping you to bring you the correct ingredient. Okay, so they will bring you the flour, bring you the egg, and bring you the sugar and whatnot, and you will be mixing it in the right order as per the cookbook. So together, these 3 types of RNA work together to allow the process of translation to occur. So let's take a closer look at the tRNA. The tRNA looks something like this. It's kind of like a clover leaf, if you would, and it's a single-stranded RNA that loops back on itself. At the bottom of the tRNA is something called the anticodon. Okay, anticodon. The anticodon is going to be complementary to the codon that is found on the mRNA. So each tRNA is going to be carrying a specific amino acid, and the amino acid that it carries is going to be the same as the codon that is on the mRNA. And so if the anticodon on the tRNA does not match the codon on the mRNA, then the amino acids will not come in. Okay, so you would have different types of tRNA, each carrying a particular amino acid, and in order for that amino acid to go to the ribosome, the anticodon on the tRNA must be complementary to the codon on the mRNA. So now let's take a look at the actual process of translation. Here we can see there is an mRNA sequence, and to keep it short, we are just going to have a few codons here. We have the AUG, which is going to be the start codon, and we have the UAA, which is going to be the stop codon. So in order for translation to happen, what you need is for the ribosome to come in. So the ribosome is going to have the large subunit and the small subunit, and they're going to clamp onto the messenger RNA. And in the ribosome, there are actually three sites. We have the A site, which is where a transfer RNA is going to bind to. We have a P site, which is where the peptide bond is going to be formed. And we have an E site, which is the exit site for the tRNA to leave the ribosome after it has brought the correct amino acid. So in this particular examples, we have the AUG as our start codon. So if you look that up in the codon chart, that actually codes for the amino acid MET or methionine, which is shown here. So remember, there are many tRNAs floating around, and which one will be the correct one to go into the ribosome? Well, you have to make sure that the anticodon on the tRNA is going to be complementary to the codon here on the mRNA in order for them to be able to dock. So as you can see here, the tRNA carrying the methionine is going to be able to bind, and you can see the anticodon here is going to be complementary to the codon. And so next what happens is we are going to have to bring in another amino acid, and UUU will be coding for the amino acid PHE, and the PHE amino acid will be carried by the tRNA that has an anticodon of AAA, because AAA is going to be complementary to UUU. So you can see the amino acid comes in, and it will enter the A site, the accepting site, acceptor site, and then the first tRNA is going to be in the P site, the site where the peptide bond is going to form. So you can see the MET, the first amino acid, and the second amino acid, they don't have a bond between them yet, but as soon as they come in, tRNA comes in, what's going to happen is you're going to be able to form that bond, and the methionine, the first amino acid, will be released from the tRNA, and then the ribosome will shift over. So now you have two amino acids on your protein, and the first tRNA is now going to be in the E site, where it will be exiting the complex here. So next we have AUA, which means we are looking for a transfer RNA with anticodon of UAU, which is shown right here, and that's what's going to happen. The UAU will come in, and then at the same time the first tRNA will exit, and everything just kind of shift over, and then the growing chain is going to be transferred to the latest tRNA. So now you can see there are three amino acids in your chain, and this is going to continue, right? We have another next one coming in, forming the peptide bond. We're gonna transfer these three amino acids to the new one that's coming in, and at the same time we're gonna have the old transfer RNA exiting at the E site, and then we're gonna keep going until we reach the stop codon, and there are no tRNA that is complementary to the stop codon. Instead, water is going to come in and break the amino acid chain from the tRNA, and you will have your protein synthesized that way. And that's the process of translation. There are approximately 42,000 genes that exist within the human body. Not all the proteins are required at all times. Insulin, for example, is only required in a cell when the glucose level is high. It would be inefficient for a cell to transcribe and translate the insulin gene when glucose levels are low. So cells have developed various methods by which they can control the transcription and translation of a gene depending on their need. Some genes are always needed in a cell, and therefore they are constantly being transcribed and translated. These are known as housekeeping genes. Transcription factors are special proteins that can help turn on a gene when it's required, and they could also turn off a gene when it's not required. So these kind of controls are called gene regulation, and gene regulations are vital to an organism survival. There are generally 4 types of controls that your cell use to regulate gene expression, and they're all summarized in the table here. So you can go ahead and read that on your own and get a better understanding of these gene expression controls. In the last part of the video, we are going to be looking at mutations. Permanent changes in the genetic sequence of an organism are called mutations, and by changing the genetic sequence, changing your DNA, the ACTG stuff, might result in changing the amino acid sequence. Right? So if you change your codon from AAT to AAC, right, that might result in a change in the amino acid sequence, although not always, because as you know, our codons are redundant, right? We have more codons than we have for amino acids. So if the change results in a codon that encodes for the same amino acid, then you will not and be changing your amino acid sequence at the end, which is a good thing. So you also know that protein functions are dependent on the order of amino acids. So if you change the amino acid sequence because of the mutation, you might end up changing the shape of the protein and therefore altering its function. Mutations can happen during any stage of the cell's life. For example, if someone use a tanning bath a lot, then the UV light might cause the mutation to occur even when a cell is not dividing or anything. But most DNA damage and changes occurs while DNA replication is taking place. There are spell checking mechanisms, as we learned, right? DNA polymerase 3 and DNA polymerase 1 can do self-checking to see if there is any kind of mistakes, but sometimes the mistakes kind of occurs anyway, and you're not able to correct all of them, and that's when you have a permanent change in your genetic sequence resulting in mutation. So we're going to look at what's called a point mutation. In point mutation, you are changing one base, and we call that a base substitution. So there are 3 possible outcomes of a base substitution, and the first one is what's called a neutral or silent mutation. So in a neutral mutation, the mutation does not result in a different amino acid. So if I have, say, UUU, let's say UUU was the original codon. Now I am doing it in the level of mRNA to make it easier to check on the codon chart, but you have to understand the mutation for it to be permanent has to occur at the DNA level, right? So on the coding strand, coding DNA, right, the UUU would have been a TTT. Okay, so let's say the TTT in the DNA is mutated into TTC. Okay, which means when you do the transcription, your mRNA, instead of getting UUU, you are now going to have UUC. Okay? So let's take a look at what happens when you translate this into the protein. So UUU, according to our codon chart, is going to code for PXE. Right? So that's the original. We call this the wild type. Okay? The wild type is PXE. In the mutant, right, when you change it to UUC, right, and we look up the codon on the codon chart, it also happens to be coding for PXE. So in other words, the mutation did not change your protein sequence, and so there are virtually no consequences, right? You're going to be getting the exact same protein as the original version, and there will be no changes in the functions or shape of the protein, so you wouldn't even know you have this mutation. That's why it's called a silent or neutral mutation. So if you're going to have a mutation, this is probably the best kind you want to have, okay? Because it's not going to have any negative effect on your body. The next kind of mutation is called a missense mutation. So let's look at our coding DNA again. Let's say the original coding DNA is going to be TTT again, okay? Which means when you do transcription, your mRNA is going to become UUU. Remember, the mRNA is exactly the same as the coding DNA, with the exception that T will be replaced by uracil. So when you do translation, the protein that is coded by UUU would be PHE. So let's say there is an error during DNA replication, and the TTT becomes TAT. Okay, so when you do the transcription, now it's going to be UAU. And if you look up UAU, UAU codes for TYR. So now this is going to be a different amino acid. So is this going to negatively impact the protein's function? Well, it's hard to say without knowing where the mutation occurs. You might remember an enzyme has an active site, right? And the active site is where the substrate binds to. So like we might have some kind of like hexagon looking thing binding to the active site. Now if this mutation encodes for an amino acid that is in the active site, then by changing it, you can change the shape of the active site to something completely different. And so the a substrate will no longer be able to fit. So in that case, it could be a bad mutation. However, if the mutation encodes for an amino acid that is in a different part of the enzyme of the protein, then it might not change the shape of the protein at all. So you know, to answer the question of whether this is a bad mutation or not, it really depends on the location of that amino acid. Okay, sometimes it could have detrimental effects, other times not so much. Last, we have something called a nonsense mutation. So in a nonsense mutation, we would have a let's put another coding strand, coding DNA. So in the coding DNA, let's say we have TAC, okay, TAC, and we're gonna mutate it to TAG. Okay, so what's gonna happen here? When you do the transcription, the RNA will have the same codon, same coding information as the coding DNA. So this will give me UAC, and TAG will give me UAG. Okay? And if we have the protein now, this is going to give me UAC is going to give me TYR, and UAG is going to give me a stop codon. Okay? So is this good? Is this bad? Well, let's say this is the entire mRNA, and the stop codon was supposed to be at the very end. Okay, and let's say the UAC is somewhere here, UAC, and after the mutation, this becomes UAG. So when the ribosome comes here and try to make this protein, it will stop at this point rather than over here. So you might have the first amino acid, second amino acid, third amino acid, and then you will stop because you've introduced a stop codon at an early point, and this is going to cause the proteins to not be created, right? We call this a truncated protein, a shortened protein. So this could be pretty bad if the nonsense mutation happens early on in the sequence. However, if the nonsense mutation occurs at, say, here, right, then you will still be able to make most of the protein, and then you might lose a few amino acid at the end. Okay, is that better, maybe. Depends. Again, depends on if this last few amino acids are crucial, right? For some proteins, the last few amino acids are critical because it's like a signaling end that tells the proteins where to go. So for those proteins, losing this last part is going to be detrimental. But for other proteins, it might be okay to lose a few, right? So to summarize, it's not that nonsense or worse than missense, and vice versa. It all depends on where it happens. Okay, so the location at which the mutation occurs is going to be important. So just to recap, neutral means the mutated codon codes for the same amino acid as the original. Missense means the mutated codon will code for a different amino acid as the original. And finally, nonsense means the mutated codon is a stop codon, and it causes protein synthesis to terminate prematurely, creating shortened proteins or a truncated protein, as they're called. Here is a summary of all the mutations one more time. On the top left here, we can see the original strands, both the coding strand and the template strand, and the supposed mRNA transcript and the proteins that you're going to produce. So if you have a mutation here, right, if the T and the A changes to G, C, and G, then what's going to happen is you are going to have a silent mutation. Okay, and you should double check this on your codon chart, right? You can look up CCU, and you will see that it encodes for proline. After the mutation, you have CCC, which is still going to be encoding for the same amino acid. Over here, if I replace the G and C here at the third position here with A and T, right, then you're going to end up with a CAC as opposed to CGC, and so the amino acid changes from arginine to histidine, and that is going to be a missense mutation. And finally, if we have a mutation over here in the middle, changing the TA to AT, okay, that's going to cause the codon to change from UAU, which encodes for threonine, to UAA, and that represents the stop codon. So instead of making the 4 amino acid long protein, you will only be having 2 amino acids, and this protein is shortened. We call it a truncated protein. So you might remember sickle cell anemia. We first talked about this in lecture number 2 when we discussed protein sequences. And here is the primary sequence of the protein in the original wild type sequence, and in the sickle cell anemia, what happens is the mutation, it's a base substitute mutation, and it causes the codon to go from a glutamic acid, which actually is a polar amino acid, and it changed it to GUG basically, right? And that encodes for valine, and that's a non-polar amino acid, and just by switching one amino acid, in this case it's a critical change that causes the hemoglobin to misfold, and as a result it alters the shape of your red cell. So like I said, sometimes the missense mutation, just one single mutation, can cause drastic changes. Other times not so much. There is another type of point mutation. We can be inserting something at a single point or deleting something at a single point. And when you insert or delete a nucleotide or nucleotides from the original DNA, you are going to potentially lead to something called a frame shift mutation. Okay, so anytime you add or delete nucleotides that are not multiples of 3, it will lead to a frame shift. So let me explain what this means on the next slide. So let's read this sentence and say we can only read in triplets, okay, 3 letters at a time. So it would say the big cat saw the fat dog, right? Now over here in this next sentence, what I did is I deleted the B. Right, so there's no B here, and remember we can only read 3 letters at a time. So now we're gonna read the, and then IGC, ATS, AWT, HEF, ATD, and OG. Right, so the entire sentence is no longer the same as the original. In fact, it doesn't make any sense. Anything after the point of deletion, it's going to be nonsense for us. So we call this a frame shift mutation. But if we delete the entire triplet, if we delete things that are multiples of 3, let's say I delete the word big, I can still read the sentence. So the cat saw the fat dog. Okay. So what happens if we add something? If we add an extra nucleotide here, right? I added a K here between the H and E here, and remember we can only read in triplets. So now you can see the sentence is all scrambled up again, and we're not going to be able to make sense of it. So anything downstream of the insertion or the deletion will have a frame shift, and it's just going to be nonsense basically. And over here we can add 3 letter words, okay? And by adding 3 letter words, we did not change our frame, right? And we can still preserve much of the sentence. Okay, so that's what a reading frame is. So in this example, you can see this is the wild type, the original mRNA, and again, right, the changes happened in the DNA level, but for simpler explanation, we're just going to focus on the mRNA here. So that's the original, and you can look up the codons, and they code for these 4 amino acids, followed by a stop. So in the first example, we have an extra U. Okay, extra U, which means on the DNA level, you have inserted an extra T in the coding strand. Okay, so by inserting an extra U here, remember your ribosomes read these codes in triplets, just like the example we did on the previous slide. So the ribosome will read AUG, okay, and so the tRNA will come in, and then it would deliver the MET, and then the next one, instead of reading AAG, because we inserted the extra U, the ribosome will now read UAA, and UAA is a stop codon, right? So by inserting something, we've created a nonsense mutation. Okay, so you can create nonsense mutations in different ways. You can create it by base substitution, where you actually change the nucleotide, or you can be inserting nucleotides as well. And in fact, you can even be deleting a nucleotide to create a nonsense mutation, and we can think about how that is possible. Next, we have a frame shift causing extensive missense. So over here, we are going to be deleting a U. Okay, so deleting a U. So this third U is going to be taken out, and you can see you would have AUG, AAG, and those are fine. They are the same compared to before. But then the next 3, because you took out the U, instead of UUU, the ribosome will now read UUG, which encodes for something entirely different. And then it will read GCU, and then something else different. In fact, the original stop codon is no longer there. So this is going to go on for some time, right? It's going to be like AA and then whatever the next one is. And so you end up having a few correct amino acids followed by a bunch of mistakes, right? That's why it says extensive missense mutation. And in fact, you're going to probably end up with a much longer proteins because the original stop codon is gone. In this last example, we have insertion or deletion of 3 nucleotides. So if you're going to have a deletion mutation or insertion mutation, this one is the least damaging, okay, compared to the other 2. Because when you take out AAG, then you do not change the reading frame. There's no frame shift mutation in this case. You will still be able to read AUG, the first amino acid, and then after taking these 3 out, you will have the UUU rather, and that's the next amino acid, and the next, and the next. So you still make up the protein, but now you'll be missing a single amino acid. And again, is that good or bad? Well, it's better than the other 2 for sure, because this one you're not having a protein, and this one you have a protein that consists of wrong amino acids for the majority of the sequence, and here you're missing that amino acid. And again, if that amino acid is not in a critical location in the protein, then maybe it's going to be okay. So let's try a few examples. In this example, it says create a base substitution in the coding strand below that will result in a missense mutation. So this is the coding And to do these kind of questions is always best to write down the mRNA. Okay, it's much easier to work with because that's what the codon chart is for. Right? The codon chart is for the codes in the mRNA. So if this is coding, then the mRNA is going to be the exact same thing, but you have to replace the T with the U. Okay, if you're still not familiar with this concept, you should go back to the previous video and take a look again. Make sure you understand the relationship between the coding strand and the mRNA strand. So here we are, and we want to have a missense mutation. So we can just pick any of these amino acids, right? Codons rather. So let's pick CAG. Let's see. So CAG is going to code for GLN. Okay, so I want to change it so that it's going to code for something else. Okay, so let's say I wanted to code for HIS instead, right? So the code for HIS is going to be CAU, right? So that means instead of CAG, if I change this to CAU, then I will have histidine instead of glutamic acid. And so if I change this to U to reflect that change in the coding sequence, this one must be a T. Okay. Let me try another one. Okay. So instead of doing that, I could also take a look at AAG. Okay, so AAG is going to code for LYS. So I can change the first one to C, right? And then that would be a C. And now CAG is going to code for GLN, right? So we change it from LYS to GLN by changing the first nucleotide. Right? So in general, if you want to create a missense mutation, changing the first one is easiest, okay? Because changing the first one will almost always guarantee you you change the amino acid. Not always though, okay? But very likely. Example 2. This is coding strand, and we want to create a base substitution that will result in a nonsense mutation. So again, coding strand, and we want to write the mRNA sequence. So this will be AUG, CAG, AAG, AAU, GAU, UAA. So a nonsense mutation means after the change, I have to have one of the stop codons. So let's write out the stop codons first for reference. So you got A in biology, you are good, you are awesome. Those are the stop codons. And I mean, if you don't remember the acronyms, you can always look it up in the codon chart here. So I want to look for something here where one change in the nucleotide would result in one of these stop codons. And so over here, we see there's a CAG. If I change the C to a U, then I would get UAG, which is a stop codon, right? So that does it. And in order for this to happen in DNA, right, this must have been a T. Okay, so that's how you can create nonsense mutation. You might notice you can also do it in the third one, right? Here instead of AAG, if I change it to UAG, right, TAG here, right, that's also going to be a stop codon. All right. In this next example, we have to create a base substitution in the template strand this time that will result in a nonsense mutation. So this is template, and so when we do the mRNA, we have to use complementary base pairing rule. So TAC will become AUG, GUC, AAG, UUA, and CUA, and UAA. Alright, so now I gotta make a change in one of these things to create the stop codon. Okay, so these are the stop codons. Okay, so we have to look here and see if there is anything I can change to make the stop codon. So over here we have AAG, so I can change the A to a U, and that would be a stop codon, right? And then the rest of the proteins will not be made. And to reflect that on the template strand, this one would have to be an A now. Okay, remember we are looking at the template strand, so to go back I must use complementary rules. Okay, we cannot just copy it like we did in the previous example. Example 4. Create an insertion mutation. So we're going to add in an extra amino nucleotides in the coding strand below, so that it will result in a truncated, a shortened protein. So we need to add in a nucleotide that would introduce a stop codon prematurely. So let's write the mRNA first. So we have AUG, GAA, UUC, AAU, CAU, UAA. Okay, so what are we going to insert? Remember, we have to insert something that will create either UAG, UAA, or UGA. Okay, so where can we insert something to create the stop codon? Well, we have AUG here. I can insert U over here, right? So if I insert a U here, then the ribosome will read AUG, and then the next thing it will read would be UGA, and UGA would be a stop codon, and the rest of it would not be made, right? And to reflect that in the coding strand, I would be inserting a T. Okay, so that's how you would do different types of mutation questions. There are more practice in the study guide, so please go through that, and I'll be doing more examples with you during the virtual sessions. But that's pretty much for it for this video. Make sure you're able to describe the process of protein synthesis, the transcription, translation, various gene regulation mechanisms, as well as identify different types of mutations. Thanks for watching.